The mic's working. Hello, everyone. I think it should all be doing stuff. Can everyone hear me? Yes? Hello? Anyway, should be starting. Sorry about the delayed start. We should... That's really weird. Okay. The reason why it's taking a while to start is because I'm trying to get a person on who is honestly one of the loveliest people on the planet. His name's Dr. Stephen Hobbs. I have interviewed him on my other channel, and he is an absolute sweetheart and a very fascinating person. And we didn't organise this until after I started the opening sequence, so we're going to try and organise that to come on, that to happen. So anyway, Stephen, could you try your Twitter again? I sent you the link again. It's a bit weird because when you enter, it gives you your visual and you can move around, and then you have to enter again. So Stephen says, thanks, I question your standards. You are lovely. I enjoyed you coming on. Anyway, so hopefully we can get everyone in. Get people all excited about microbes and stuff. Anyway, so I would like to say a very quick hello to everyone who've joined and everyone who's going to watch this in the future. Hello and welcome. MathPig is first because the host doesn't count. Notcha, hello. Mofab, hello again. Stephen, Pale Blue Mama. Hello, Mofab. I need to mod you on that one. Ian Burns, hello. I am the woman who is both too old and too young to have an opinion on the internet. I think, honestly, that's probably because I have one thing that is not okay for me to have, and that's a penis. I don't have that, so therefore I'm not allowed to have an opinion according to some people. And also hello to Ebony the Burmese. Foppish Dilettante, word up. And yes, the stream is starting. And Vagrant Sam, hello, and stuck in Florida. So, hello everyone, and Dottie Little, thank you for coming. Stephen, I can't see you on StreamYards. I don't know what's going on. Oh, there you are. Oh, he's here. Hello. Hello, hang on, I will very quickly... give me a moment. Can you say something?
How are you?
I'm good. Hello. I can hear you, and I hope the chat can hear you, but they can't see you because I haven't set up OBS to be able to see you. Hang on.
No worries.
Hang on, I'll go onto my other StreamYard setup. You are not Telltale, by the way. Everyone, this is Stephen Hobbs. Hello. Anyway, who are you? What are you? How are you? And why are you?
I don't know any of those answers. Stephen Hobbs, I'm on Twitter. That's what I do, really. I'm the division director of STEM and all kinds of other stuff as well. However, I focus mostly on science and mathematics. I study everything, really. I've been a professor of chemistry, biology, and physics. So I've taught everything from astronomy all the way down to microbes. So depending on what size you want to talk about, I've got it.
That's part of the reason why I wanted you to come on, because you are a bit of a microbe nerd along with myself, and microbes are very cool. So they're very cool. Have you been watching any of the videos, other streams that I've been putting on at the most inconvenient time for you?
Three. I've gotten 3 all the way through. I've gotten a couple of others that have either started and I've got ready to go, or I have not gotten to them yet, but I will get to them.
Fair enough. So I'm glad to have you on. This paper that I have chosen is a little bit of a continuation on from last week's when we were talking about the microbes in the mouths of snakes. And this time we're talking about microbes in the mouths of wild-caught snakes, of venomous and non-venomous Indian snakes. So snakes from India. So that's, yeah. So I thought we'd probably do that. The problem that I'm having with this particular project online is that I'm seeing some really fascinating papers, but they're all about 20 pages long. And so I can't exactly read a 20-page long scientific paper because that would be like a 4-hour stream, and no one wants to listen to me talk about science for 4 hours. Even I wouldn't want to do that. So that's one of the problems. So I'm really grateful to have you on. Again, you are not Telltale Atheist. That was the last person who I had on from last time. And you are also not Scientist Mel, because Scientist Mel was on last week and we read the paper together.
She is much better looking than I am.
Oh, I don't know. You're pretty good looking. It's got, the glasses. It's just beautiful. They're gorgeous. All right, so, oh, hang on. Oh, geez, where am I? Okay, so I'm going to click over. So, and I will do, okay, hang on. That's Vermilion paper. And so what I'm going to do is, again, we did not know this was going to happen, so please, I do apologize.
No, you're just fine for me.
Yep. Display capture. Nope, not that one. So would you be able to entertain the chat while I struggle with OBS, if that's okay?
Sure, absolutely.
Thank you. I appreciate that. So where did you what's your favorite food at the moment?
I love basically anything Indian, any Indian food. Just give it to me. That's one of the things that I cannot find where I live in the middle of West Texas. We don't have any, so I've learned to cook it.
Ooh,
which sounds weird, but it's a staple in my diet at this point. So, yeah, I am learning how to use food I have never even heard of before.
Ooh,
so one half of my family is Swedish, and the other half are Creole and Cajun. So I've learned how to put those two things together, which is terrible, but it works. It works for me. But once I got a taste for Indian food, I just fell in love with it.
It is good. I have a really amazing butter chicken recipe that I will give to you if you remind me.
Absolutely. That's what I had tonight, actually. Yes.
Ooh. I don't think I have your Twitter profile picture, so you're going to have to go without being having a picture, unfortunately.
Oh,
I'm sorry. Okay, I think that's it. I think that's it. All right, so we're all set up. Okay, so today we are going to be talking about the next generation sequencing analysis reveals high bacterial diversity in wild venomous and non-venomous snakes from India. I will try and send this through to you right now.
Was that the article that you had posted on YouTube? Yeah, I had that. Give me 1 second, but go ahead. You can go ahead and send it to me.
Okay.
I should be able to.
Actually, what I'll do is I will send you. It's open source, so you don't have to find it.
I've got it.
I'll just send it. Oh, you got it? Oh, gosh, you're good.
I got it.
So, so this is what we're going to be talking about. And as you'll probably know, I'm just going to be reading some of it and you can jump in or we can talk about it. Yeah. So basically the background says the oral back cavities of snakes are replete with various types of bacterial flora. Culture-dependent studies suggest that some of the bacterial species are responsible for secondary bacterial infection associated with snakebite. A complete profile of the Ophidian oral bacterial community has been unreported until now. Therefore, in the present study, we are determined to determine the complete bacterial compositions in the oral cavity of some snakes from India. That's basically it.
Well, this is one question that I have just right there from what showed up was my question is, are those exogenous or endogenous, and are they absolutely confirmed? Exogenous meaning coming from the herp itself as compared to endogenous from the herp itself, the snake, as compared to exogenous, which would be on the person's skin or wherever they bit.
Yeah, well, the last week's one was they were thinking it was exogenous, meaning because they were thinking, oh yeah, the bacteria in the snake's mouth is going to be from the fecal contamination, and they found that the fecal contamination of the mouths of the snake was almost non-existent. So it was endogenous. Okay. Yep.
Okay.
And so the methods was they just basically used DNA analysis in PCR. Oh, hello, Mr. Doctor. How are you going, by the way? Good friend of mine, everyone. And so basically the DNA they just did PCR amplification and looking at the 16S RNA gene using the V3 region-specific primers. So could you explain what the V3 region-specific primers are?
Not all the way, but I can explain what they are, yes.
Yeah.
Because each one is going to be slightly different per species. So what happens when you have a primer, you are looking for a specific spot where DNA starts to replicate. So we've got this primer that's set up of RNA, and it is specific for that particular strain or that particular DNA sample. So you would think, well, DNA and RNA don't really fit together. Well, they do to begin with, because eventually that RNA will be removed and replaced with DNA, which means it's got the particular code on it. It's easy to separate, and at that point we can then measure it.
Yep. Yeah.
Okay.
So it's kind of like what would be a good analogy for that? It'd be like a key, kind of. You know how a key,
yeah.
Mm-hmm. Yeah. So like a key has specific grooves, and if it doesn't fit the lock, it won't. Like if the grooves don't match up, the key won't work, and it's something similar. Correct. It's the grooves in the key. I think that's probably the closest we could get.
Yeah.
It's not really a lock and key in the sense it unlocks it. It's more that the grooves in the key needs to fit with the grooves of the lock to be able to function. It's that part of
Absolutely correct.
Oh, and Almost a Doctor is trying to make a quick video on rats. That'd be interesting.
Nice.
And okay, so we'll go and talk about the results because a bit later on, so we won't talk about that. But it was really interesting that it said our study reports that all the unique and common bacterial species found among oral microbes of snake studies, while the majority of commonly abundant species were pathogens or opportunic pathogens to humans. A wide difference in Ophidian and oral bacterial flora suggests variation by individual species and geographical region. The conclusion is that the present study would provide a foundation for further research on snakes to recognize the potential drugs and antibiotics for the different infectious diseases. So these, as much as what people are going, oh, why are you just growing bacteria? Like, why would we care? Well, the thing is, this sort of stuff can make animals, like, can help us solve things like antimicrobial resistance. which is
Absolutely.
pretty cool. All right, so I will go turn that off. Oh, and then I'm going to scroll down. Yeah, all right. So the background, vertebrates form mutual relationships with huge and complex microbial flora that inhabit their gastrointestinal tract. A major proportion of these microbes probably assist in essential processes of energy and nutrient acquisition in the host. The combination of next-generation DNA sequencing methods, ecological aspects, and bioinformatics analysis tools is rapidly expanding our comprehension of the evolution and function of vertebrate-related bacterial communities. The diet and genotype impact the bacterial diversity since the bacterial communities co-diversified with their hosts. Most of the studies have tended to characterize fecal microbiomes from captive animals, often from laboratory or zoos. And that's a problem.
Yeah. It's also easy to collect. That's why they do that.
Yeah. I mean, it's pretty easy to... Yeah. I was reading a paper that was, you know what it's like when you read a paper and then all of a sudden you're going down this massive rabbit hole. I was with Notcha the other night and I read where the intestinal tract of a human can communicate with the microflora of the intestines if there's been any damage done to the intestine. And I thought that was crazy. Like that interspecies communication on the cellular level. I was just like, oh, it was so incredible.
Absolutely. And then the fact that they evolved together and separate at the same time is even more mind-blowing to me. Looking at that from an evolutionary standpoint is phenomenal.
I totally agree. I absolutely agree. I also want to say a quick hello to .nerd for popping in. I do think you were slightly beforehand, but you were on a different account. But thank you for popping in, Snake. .nerve. And okay, let's continue on. So most studies investigating evolutionary patterns in hang on, where are we? All right, I just missed. Most studies investigating evolutionary patterns in vertebrate gut microbiome. And let's go. I really should get somebody to do this for me. On mammals, on mammals and birds only. Until now, very few studies have analysed the gut microbiome of squamate reptiles, of snakes and lizards, despite this being one of the most diverse and successful vertebrate clades.
I want to add this. I still think we should look at the tuatara.
The tuatara? That's
absolutely
a bird, isn't it? If I'm correct.
No, tuatara is a native reptile. It's a lizard-like reptile, but not a lizard. From New Zealand.
Oh.
My favorite reptile of all time, the tuatara. I mean, this is really old physiology. So if we're going to look at how long they have been around, I mean, compared to the rest of us, they've been around forever. So it's really old physiology. I want to find out even more about how their microbiome works with what they have, of course, and work in different areas of New Zealand at the same time. Yeah.
They that would be interesting. It would be very interesting because they would probably have microbiota that only exists in that particular species.
Absolutely.
That's something that we don't really talk about. Is like every time a species goes extinct, all of their a good chunk of their parasites go with them because they are specific to that particular species. So when a species goes extinct, you're probably also losing about 3 other species or more of parasites. And
very easily.
Yeah. And that's kind of sad, really.
It's really sad.
Yeah. Okay, so presently the use of reptiles has increased in the investigations of infectious disease, comparative That's the word, physiology. I cannot read, honestly. Well, I can read, I can't speak. Stem cell experiments, evaluation of phylogenetic relations with birds and other vertebrates, and therapeutic drug development. Among the reptiles, snakes have been utilized for the isolation of different types of peptides from venom for numerous purposes, which is very cool.
Can I throw something in? It's just a small correction. It drives me crazy in the healthcare field. It's antivenin rather than antivenom. It's not even in this article. It's not, but I know that it shows up so often. And the vial says antivenin on it because that's how it's supposed to be. But people will read that and say, well, that's not antivenom. The whole time the patient is waiting for the antivenin to be administered.
Right.
But they're arguing over that. Oh my God, it drives me crazy.
So why isn't antivenin not antivenom? Why is it? It
is an antivenom. However, it is a protein. It's an anti-protein. Well, protein's in with IN.
Ah, okay. Ah, all right. Learn something new every day.
It's one of the few things I'm pedantic about. Like like you've seen the way that I write. It's terrible. But it drives me crazy. And I don't know why. It's something that's always been. I've learned this, and I've seen people in the healthcare field look at it. They can read it. They know it. They can see it. It's staring them in the face, and they still ignore it. What happened? Where did the disconnect come in?
I don't
know. Go ahead.
All right. Well, thank you. I am glad to know that. So next time if I'm ever bitten by one of the deadliest snakes on the planet, I'll be able to go, make sure the person who's administering the antivenom is actually aware it's antivenin. I will do that. Thank you. I appreciate that. Okay. Microorganisms, including bacteria and fungi, or fungi, or however you want to pronounce it, because he's a fun guy, naturally inhabit the oral cavity and gut of snakes, and actually everything that has a mouth, really. So you don't have to worry about that. That's why being bitten by a human is significantly worse than being bitten by a dog.
You got it.
The literature suggests that oral cavities of venomous and non-venomous snakes are colonized by numerous species of anaerobic and aerobic bacteria. Since the Ophidian oral bacteria may be inoculated during a snake's bite, bacterial multiplication and infection may occur under favorable conditions. A strong connection has been established between microorganism presence in the abscesses or in patients' lesions and those from snakes' oral cavities. Because I'm so good at doing this. Okay, so anything you want to add to that?
I don't know if anybody really, I don't know their background, so I don't know if they know why there's bacteria in their mouth anyway.
Well,
so we'll go ahead with that.
Well, may as well explain why. Yeah, we should probably explain why.
So most people have, I'd say most organisms with a mouth have bacteria in them. That bacteria has found a niche where they can survive. Well, that niche can be beneficial to the host, or it can be, a detriment to the host. Either way, they're either going to fight for that area, such as the teeth, the oral cavity, of course, or the host is going to try to supply everything that bacteria needs because it benefits them. Example, we have bacteria in our GI tract that provides vitamin K. Vitamin K is used for clotting, so we use that. It's a benefit for us. So we basically pay the bacteria to stay there. We feed them what they want, and they benefit us greatly. There are others, bacteria that grow in our mouth that causes tooth decay. Well, that tooth decay is a bad thing, so we want to get rid of that as much as we can. That's why we brush our teeth and take care of oral hygiene, of course. There are other microbes that are in there. They're just kind of hanging out. They're just there. If they're there, great. And if they're not, oh well. We call those transient microbes. Typically, they're there temporarily, and then they'll move on. So, what we are looking at in this article, all of those are permanent bacteria. Those are bacteria that are not transient. So they actually live on and in the host like they're supposed to, because they're providing either something that the host wants, or they're providing something that the host does not want, but they're not going away anytime soon. With venomous and non-venomous snakes, or in any reptile, really and truly, you want to protect the teeth, of course. Those bacteria are there to protect the teeth because they can't brush their teeth. So we want to have some microbial antagonism there to make sure that the oral cavity stays healthy. In a venomous snake, they bite and they inject the venom so that they paralyze the prey that they're eating. We happen to just be in the way. Most venomous snakes do not want to eat a human. Like they warned you, they tried to tell you, leave me alone. You didn't listen. They bit you anyway. Unless you're walking along, you get bit in the leg from grass or something like that, of course. Most snakes don't want to interact with us. When they say that they're just as scared of us as we are of them, that's really and truly, a fact. Well, the bacteria that's in there is supplying their own health. The bacteria have found a place that is beneficial. So they are not affected by the venom, apologize. They're not affected by that. They're not affected by most of the pathogens that particular will carry around, which is good. But they have found a niche where they can fight it out with everybody else and kind of eke by a living over and over and over, being passed from one generation, one species to the next.
Yeah.
Go ahead.
I was going to say, creating venom for a snake is extremely energy, it's very energy intensive, which is why with the snake milking places, most snakes you're only supposed to feed them once a week, once a fortnight at fully grown. The snake milking places, they feed them like every week because they force the snakes to produce too much venom, and that requires a lot more food. And venom also can be part of the digestive process as well, because they have a lot of snake venom has a lot of myotoxins. So a myotoxin basically breaks down muscle. So there's a lot of myotoxins in the cocktail of the venom, which is pretty interesting. It's really interesting. So if you get bitten by a snake, there is a good chance it will try not to envenomate you because it knows that you're not edible. You're a little bit too big to eat. So yes, that's interesting. It's a very interesting part of snake physiology and psychology, I suppose you could call it.
Yes, absolutely.
Yeah. Okay, so where was that to? Snake bite. Have I read that bit? Where was that to? Snake bite generated secondary wound infections. Have I read that bit?
That's where we are. No, that's where we are right now.
All right. Okay. Involve a polymicrobial mixture of microorganisms originating from the ophidian oral cavity. Bites from non-venomous snakes can also cause injury. That's true. Yeah. As a result of lacerations by the snake's teeth and subsequent infection. The identification of snake-associated microorganisms is imperative to extend our insight into these life forms that inhabit the buccal cavity, and furthermore to acquire understanding of the etiological operators of secondary infections resulting from accidents during handling. I was able to read something then. It's a slight miracle. Hence, the profile of microbial vulnerability to antimicrobials must be investigated to encourage the development of adequate treatments of human accidents and snake bacterial infections. It has been suggested that the oral microbiota of the snakes reflect the fecal flora of their ingested prey, since these sufferers frequently defecate in the moment they are being ingested. And that's what I talked about last week with Mel. However, recent culture-independent high-throughput sequencing studies identified that bacterial taxa present in the oral cavity of snakes were distinct from fecal microbiota of their prey. And
that's where the microbial antagonism comes in.
Yeah. Yeah. So microbial antagonism is basically the bacteria that's already set up goes, no, fuck off, and then kicks them out. That's basically it.
Exactly.
And that's why some microbes we can use to treat infections like lactobacillus species such as the stuff that you put in yogurt can be used to treat thrush, because thrush is caused by Candida albicans, if I'm correct.
Right. Yep, you're correct.
And because it's Candida albicans and Candida cerevisiae, isn't it? Or am I thinking of something else?
Right.
Yeah. So the albicans is the parasitical one stuff, and the lactobacillus is antagonistic towards Candida albicans, and that's why if you have a thrush infection, it's probably just cheaper to slather it with yogurt than going to the chemist. Although that is not what I suggest you do. If you do have a thrush infection, please go and see the doctor. But if it's like 1 o'clock in the morning and you're itching like nothing else, and you have yogurt in the fridge, you could try that and then go and see your doctor. So, all right, where are we up to? Despite the influence, I think, however, recent culture dependent, of 23, so we're up to 23. Despite the influence of association of bacteria in snakes and the influence of these bacteria in humans, there are a few studies on the characterization and distribution of these microorganisms. Next generation deep sequencing of hypervariable regions from 16S ribosomal RNA genes is a useful tool for understanding the microbial communities in several organisms. Recently, a metagenomic sequencing study on the timber rattlesnake has unveiled the complete gut microbiome that is, and let me turn that off, and go all the way back up here. It's essential for the health and nutrition of the species and the microorganisms associated with disease transmission between the snake and other animals. However, complete snake oral metagenomic sequencing has not been reported until now. Therefore, the objective of the present study is to identify the bacterial community diversity in the oral cavity of 2 venomous and 1 non-venomous species of snake native to India. We'll read the microbial, the sampling. We can have a chat about your thoughts on this. So how you think they went about. So according to the availability of total of snore fakes, 4 snakes from 3 species were used in this study. Live venomous snakes, namely 1 Indian cobra, Naja naja. I actually knew someone called Naja.
Really?
And 1 king. Yeah, I did. Yeah. Apparently Naja is like I think it's the ancient Egyptian word for snake. I don't think it's Latin, but it could be Latin. I don't know. And 1 king cobra, Ophiophagus hanna, and a non-venomous Indian python, Python molaris, were captured from the wild from the Wayanad district of Kerala state, India. The snakes were handled carefully, I would hope so, throughout the exercise with the help of snake handlers and experts. Upon capture, each snake was transported immediately to the laboratory, whatever, how you want to pronounce that. Potato. Soon after the arrival of snakes, oral samples were collected. The animal handlers secured the head while a veterinarian opened the mouth of the snake using a sterile wooden spatula. Oral swab samples were collected from each snake using commercially available sterile cotton swab sticks. After collection, swabs were placed separately into sterile tubes and transported immediately to the lab on ice. Snakes were released back into the wild after the exercise. A fresh road-killed king cobra presented in a veterinary hospital near the laboratory was also used for sample collection.
Use what you've got.
Yep, pretty much. Okay, so I'm just having a quick look at the chat. Going back from so Notjo is talking about his pet, right? Asking if isn't the saliva poisonous. I don't know.
From the rat?
From the rat. I don't know. Maybe.
Is it from the rat now? I mean, it shouldn't be poisonous.
Hopefully not. Ian Burns says, I have eastern brown snakes in my backyard. Super deadly, but they generally scatter. Yeah, you don't want one. I almost stood on a baby brown snake, but luckily I had boots on and it was not that. I was like, oh shit. And it was like, oh shit. And it ran away and I was like, and then my boyfriend at the time was like, oh, the reason why you had to go was because, you had boots on. You care about our safety. And I was like, you bastard. And Notja says that he used to have eastern browns where they grew up and inland taipans, king browns. It was like deadly snake lovers heaven. And where else are we going? Notcha does say, stop treating your guest like a tutor nerd. It's like, you guys don't want to listen to me. Who wants to listen to me? When you've got the ever lovely Dr. Stephen being able to explain shit to you.
We're all here to just...
Well, I'm just the host. Okay. So you've got the DNA isolation
you get far more
amplification.
work than I do.
Pardon? What was that? Sorry.
I said
DNA...
you do far more work than I do.
Hopefully, I'll be able to get into the lab this week, but trying to pin down a time to be able to do it is proving to be a little bit more difficult than I thought it would be. But hopefully, this week I should be in the lab. But I do want to say the DNA isolation, amplification, and sequencing. I am not going to read this. If you want to read this, you may pause it and just explain it to me later in the comments below, because I'm not reading this to you because I can't read at the best of times. But can you see what I'm talking about, Doctor?
We can absolutely see.
And does that make about as much sense to you as it does to me?
Well, I mean, I do this, so I do have a question though. Why did they go so high on something from herps? They brought it all the way up to 98 degrees C.
98 degrees C?
Yeah, if you're in a reptile, I mean, the optimal temperature for those, I mean, obviously it's going to be outside, but it's going to be somewhere, 30 or less C. That'd be optimal temperature for those bacteria, unless they're extremophiles, because they're living inside a reptile, so they're not.
I think that's just the PCR cycling. I think that's just the PCR cycling. Is that what you're talking about?
Yeah. Why would they need to bring that all the way up?
For the denaturing?
Yeah.
Wasn't that what they normally do?
Well, human microbes, absolutely.
Mm.
But all microbes need to be up that high. So I wonder if they completely denatured too much or not enough. Does that make sense?
Maybe.
That would be a test. That would be more of a lab test, for the PCR than what we're talking about. So I'll stop there.
I thought that was just like standards for PCR.
Yeah, but my microbes are never all standard. I wish that they were. They're never standard.
Of course they aren't.
I mean, you'd have to just think about an archaean. If you've got an archaean in there and you try to do it at 98, it's going to laugh at you.
Mm. Yeah, I suppose. I mean, if it's a bacteria that's used to living at like 1 or 2 degrees Celsius, you probably don't need it to go up so high, because if you did, would you end up absolutely destroying the... okay. Well, I suppose India does get up to about 40 degrees C, like the ambient temperature, so that might have something to do with it.
I could see that. Okay.
Yeah. That's just my hypothesis.
The stuff nerds worry about.
Yeah. All right, so I'm going to go into the next page. So that's two of them. two of the snakes, and there's the other snake, and you can't read it because I haven't set it up because I'm absolutely horrendous at OBS, but it says the photographs of the venomous and non-venomous snakes used for oral sample collection. So the first one, A, is the king cobra.
That is terrifying.
Yeah, they're pretty scary. B is the Indian python. That looks a little bit like a what are they called? Boa constrictor. Actually, that looks a little bit like a boa constrictor. And C is the Indian cobra, Naja Naja. And D is just the oral sample collection from a snake using commercially available sterile cotton tip swab sticks. So they just kind of shove it right down the snake, and the snake's going, oh, because they never enjoy that. So and you get to see pretty pictures. There's more stuff, and even more stuff to be reading that probably doesn't make a lot of sense for the vast majority of people. So I'm not going to read that because I don't want to bore you.
Yeah, they'll all go to sleep and hit a guardrail because they're driving.
Yeah, probably. I don't want anyone to die. Ian Bird's laughing at explain shit to you. Yeah. Okay. It's like, oh, and then Notcha replies, that is precisely what nerd does often. Hey, I'm surprised we aren't talking about poo today even. Look, dude, if you want me to talk about poo, I can. Like, it's all right. Okay. There's a saying, good friends talk about men, best friends talk about poo.
Okay.
It's kind of true.
God, no. It is. Yes. All right, then. Never realized that until right now.
Okay, so the taxonomy, profiling, and community analysis of 16S RNA amplicon sequences. So I did explain what the 16S RNA is, but would you like to explain because everyone would rather listen to you?
No, go ahead.
Okay, so the 16S RNA is basically a highly preserved section within a microbe, and that's usually to do with the ribosome. So it's the bottom part from what Mel was able to pick out. So if you look at a ribosome, it kind of looks like a hamburger bun. And so you've got a fat top bit and a kind of skinny bottom bit. And through it's sort of a ribosome basically reads different codons and tells, it basically looks like that. And and the pen is like the RNA strand. And so what the ribosomal RNA figures out is it reads it and goes, okay, these three particular codons, these U, A, or G, or C, UACG, yep, because DNA is TACG. So the UACG tells the RNA, the ribosomal RNA in lots of 3, what amino acid it is saying it wants to produce, and the ribosome goes, oh, you want a tryptophan, or you want I'm trying to remember some of the names.
Isoleucine.
Yeah, all of them. Yeah, there's 20 main ones that escape my brain right now, and it goes, all right, you want tryptophan or a leucine or a cytosine or something, and it just chains them together and produces a protein. So if you have a problem like a mutation within the 16S RNA section, that means that the ribosome doesn't function properly. And then natural selection would wipe out because the proteins aren't being properly made within the microbe. And so that's why it's highly conserved. However, not all, most mutations are actually silent. Because remember how I said that you've got the three things, the codons, the three UACG in a row produces that amino acid. But if you change one, it can one of the protein, like one of the letters, and it's just a point mutation, so it just swaps out one of the letters. The chances are it's probably going to stay to be the same amino acid that it codes for. And I probably should do a video on this because it's pretty interesting. And so basically, it's like being able to swap out a letter and still mean the same thing. Like you can, if you have a sentence and then you swap out a synonym, the sentence is going to be the same. Like you are really pretty or you are really beautiful means the same thing effectively. So swapping out one of those codons means that it goes from pretty to beautiful or attractive or whatever. And so it just goes, oh, it's the same thing. And uses this, takes the same amino acid. And that's where we can track throughout the mutations, within the 16S RNA. They can track, how much it's evolved, comparing it to other microbes. So it's really, really important, because one, it's highly conserved, but two, it makes a good timeline. Mm-hmm. Because if there's a mutation at a certain point, and at that certain point, and it then multiplies and multiplies out, and then you start getting new species, we can go, oh, at that particular species of bacteria has that point mutation, but this one doesn't. So therefore, it got that point mutation after splitting from here. So it's a good time machine as well. So that's what a 16 is. Yeah. So if anyone's confused about that, let me know, and I'll try to explain it with a little. Oh, thank you. I appreciate that. Okay. So initially, the raw sequencing reads were checked for quality, adapter timer, and duplication using FastQC. And oh gosh, it's still lots of. And give me a moment. And so FastQC V0115, where the adapter trimming was performed using an in-house Perl script. So does that make any sense to you? No? Okay.
No, that's very specific for that particular study.
Yeah. So the adapter trimmed reads were merged to make V3 consensus faster using the Flash program with default parameters. So Flash is like a database of sequences. So it's basically the control F of particular sequences.
Yes.
So if you have a 30 sequence long chain, and you're like, oh, and it's all like all these, it's in a particular order, you can just go control F, and it will be able to tell you what the chances of that particular microbe belongs to. So it's basically, as I said last week, it's the control F of the of your sequences.
And it's growing, by the way. It is growing exponentially, and I love it.
It's a very useful thing. It's very useful.
Yeah, I don't know how anybody did this in it started to develop in the 70s, really, but how anybody did it in the 80s or 90s, I have no idea how they would have done that. Can you imagine the reams of paper they used to save that?
Wow. It's incredible.
It's mind-boggling that they actually did this back then. Anyway, go ahead.
So, okay, I think basically, the pre-processed reads were clustered into operational taxonomic units using. They're just still talking about how they found it. So basically, using using programs and their search parameters and things like that. It's like, for example,
they cheated. Basically, they cheated. They didn't have to go through any of the lab work to find out what bacteria they had.
Yeah, bastards.
Took a sample, mixed it all together. They didn't isolate most of the bacteria. They just put it in the program and found out what happened. Yep.
That's pretty much it. This tells you the statistical analysis, so the phylogenetic investigation of community by reconstruction of unobserved states, or PCRUST. Analysis was employed to study the functional gene profile of metabolic pathways among the samples. The metabolic profile was further analysed using the software package Statistical Analysis of Metagenic Profiles, or STAMP, version 2.0. Statistically significant p-values were calculated based on Fisher's exact test method using Story's false discovery rate method of multiple test correction within STAMP, considering p-values of more than 0.05 for comparison. So could you explain a little bit about that? Does that make any sense to you?
It does. So what we do when we're trying to compare something is we want to make sure that as much of the genome as possible is found. You only want a very small amount that's missing. And what that does is allows us to look at the metabolic pathways that are showing up to show exactly what should be where it's supposed to be. Then we can compare that. So when we compare those, everything seems to just line up like it's supposed to ensure that what we thought was the particular bacteria that should be there was actually there.
Yeah.
Does that make sense?
So, yeah. So the metabolic pathways is things like methanogens and things like that.
Glucose into energy.
Yeah.
Yeah. Glucose into energy. How do you turn, an amino acid into protein? Really?
Yep. Yeah. It's how it functions, really. How it eats and how it does its eating. So the results are showing. So the results you'd think would be the most interesting part of the paper, but generally it's not. That's what I've been learning. So the results are saying the sequencing of Pardon? Sorry? Oh, you there? You there? Yep, all good?
Sorry, it's cutting out.
Okay, yes, I can still see you. So,
okay.
Okay, so.
Okay, now you're back.
I'm back. Okay, cool. So oral swabs from the snakes were collected. So the next generation sequencing of partial 16S RNA genes based on taxonomy profiling employed in this study inferred the bacterial diversity in the oral cavities of 3 different Indian snakes, namely the Indian cobra, king cobra, and Indian python. Oral swabs from the snakes were collected, and the V3 hypervariable region of the 16S RNA gene of the microbiome was sequenced in the Illumina-based method. Total readings of 1,155,180,000, 1,085,952, 1,065,065, 1,404,902 were obtained from the cobra, king cobra, and fresh road-killed king cobra and the python samples, respectively. The average GC content of all samples were 52 to 54%, and an average base quality Fred score of 93 to 97%. After pre-processing, the V3 sequences for each sample were generated. The sequences obtained from each sample were first pulled together and then clustered using the program uClust available in QIMI version 1.8, with similarity cutoff of 0.97. From a total of 46,907 OTUs, 24,233 singletons OTUs, or less than or equal to 1 read, were removed, and 22,674 OTUs were considered for further analysis. Okay, would you be able to explain anything about that, or was that just as much jumble as... Because a lot of this is like, when you read these things,
so
yeah.
What GC content is yeah. It gets confusing very quickly unless you're versed in it.
Yeah.
It's kind of like reading a foreign language, really.
It is.
So GC is a guanine cytosine relationship. So what we try to do is find out if a particular bacteria has greater than a certain amount. In microbiology, we usually use 50%. If it's greater than 50% G to C or less than 50% G to C ratios, and that shows that they're related to each other, talking about the microbes themselves, not the snakes.
Yep. So guanine pairs with cytosine in the DNA strand, for anyone who's a bit confused. So you should roughly have about a 50%. If you sample a pure DNA sample, you should find 50% cytosine to 50% guanine. So yeah. So that's roughly, hopefully, what you should be finding. So the average GC content of all the samples were 52 to 54. Hopefully. Not all the time, because of course you've got mutations and things like that. But it should roughly be that.
So and those are all high G to C ratios.
They're high. So you
those
want
are all 50... high G to C ratios, which means the bacteria that they use are all related to each other.
That were what? Sorry, you just cut out a little bit.
Sorry about that. It shows that the bacteria are all related to each other. Because if it was a low G to C, we would have one particular type of bacteria. And if it was a high G to C ratio, then all of those would be in another family. Well, all of these are high G to C ratios, so they're all in a different family.
Okay. So a low would be 50-50, or a low would be 25-75?
25. Definitely. Right. Low would be 25. So we use the 50 as a cutoff. Anything higher than 50 is considered high. Anything lower than 50 is considered low.
Okay. So average base quality Phred score of 93 to 97%. What is a Phred score? I haven't seen that. PHRED.
Yeah, those are scores to show exactly when we're doing an analysis, what is the likelihood that everything is there. So, 93 to 97% is phenomenal. So what we had expected for that particular sample, once you find out with your G to C ratios what it should be, you then compare that to the Phred.
Okay. Okay, so here I'm going to go into the part where I'm actually kind of interested in, because we're talking about the taxonomic profiling of metagenomic sequences. So the taxonomic classifications of OTUs were carried out using the RDP classified against green genes and silver 16S RNA gene database. The relative distribution of phyla, genera, and species between the samples is shown in figures 2. The 9 phyla, including Actinobacteria, Bacteroides, Proteobacteria, Chloroflexes, Firmicutes, Cyanobacteria, TM7, so Candidus Saccharibacteria,
Saccharibacteria, yeah.
Acidobacteria, and Geminatimonadites. Geminatimonadites. Why?
Right.
Why?
Yeah, well, yeah.
So, any of
That's
those...
how you win at Scrabble, right there. Words with friends, I've got you.
That's not a word in English. No, you're right. Are there any of those phyla that you'd want to expand a little bit on? So, Actinobacteria, Bacteroides, Proteobacteria, Chloroflexi.
I deal with Proteobacteria all the time. Yeah. So,
So what's a proteobacteria?
Proteobacteria I deal with all the time. I grow them. Proteobacteria are a very basal group of bacteria. I call them the generalists. There's pretty much nothing they can't do. We just haven't figured out how to make them do it for us.
Bastards. Do you know anything about...
They have all the secrets. They just won't tell us.
How could they? Do you know anything about Gematinodates?
Hey, I have a question. Is it possible?
What was that?
Nothing.
No?
Sorry.
You don't know anything?
I would like to know more about that, and I will at this point.
Okay.
I would like to know more about it, but I know absolutely nothing about it at this point.
Yeah, no, this is the first time I've heard of Gematinodates. I don't know them either. So, and going all the way back up. Okay, so results demonstrate that proteobacteria, so the generalists, the ones, the fairly basic ones, are identified as the most predominant phyla associated with their snakes analyzed. That's not really surprising though, is it? At the species level, according
so all of these are soil bacteria for the most part, and same thing with I'm just going to call it the Gemma. Same with Gemma, and then from what I just did, just a short search, it is very understudied. So that's why neither of us really have recognized it before. There's only 2 classes of it.
So
thanks, birdies. Appreciate it.
Thank you. All right. Apparently, the stream is pretty choppy. Hopefully, I don't know if I can fix that. Okay. Okay. Yeah, I can't. I'll see if I can't change the latency. Unfortunately, sorry, everyone. Okay, so at the species level, according to the OTU-based relative bacterium angustum, Streptococcus luteaceae, Prevotella Escherichia coli, I know that one, Streptococcus algalactiae. Do you want to do this for me? Or are you just enjoying watching me die?
Agalactiae.
There we go.
Streptococcus agalactiae. these are fun. Corynebacterium durum, Bacteroides fragilis, Propionibacterium acnes, that's acne, Photobacterium damselae, that's one that we find in like dragonflies. Those are all found among the samples.
Oh, okay.
Yeah.
I know.
Sorry, I don't get to say that with an Australian accent, so they'll just have to deal with it.
That's all right. That's all right. At least you can say it better. I can say it badly. But at least I say it with an Australian accent. Most people here would be true. Escherichia coli, I think most people would know that's found mainly in the gut, but can also be found on the skin as well occasionally. Okay, so and the complete taxonomy annotation summary is displayed in additional file. Sequences that did not associate with any known reference taxon were classified as unknown or novel hits. A total of 96% of unique OTUs are reported as unknown at the species level since there were no hits. Is that normal? Is that reasonably normal?
Yeah, when we do an oral swab, you're very easily going to find 3 or 4 that we haven't seen. Yes. They released a study about 5 years ago now where they did swabs from the umbilicus, so belly buttons, and we found another 400 bacteria just from doing swabs on people's belly buttons, because really and truly, I mean, unless you have sex with somebody, you don't really come in contact with anything like that. Like,
That is
when was the
true.
last time you came in contact with a stranger's belly button? So it speciates very easily. So yeah, when we do an oral swab, I mean, when was the last time two snakes kissed?
Probably never.
So yeah, we get novel bacteria all the time.
Oh, okay, cool.
Yeah.
So it's more like if
So
you
once
find a
they hatch,
novel.
if it's a snake that lives 4 to 7 years, there's all kinds of bacteria.
So are you saying that like finding a new species isn't surprising, but if you found a new genus, that'd be sort of something to celebrate? Yeah. And if you found a new,
Absolutely.
if you found a new family, you'd be like, oh,
But if you want something named after you,
that you'd find a new family.
yes, that's what we're looking forward to.
That'd be so cool. That'd be really, really cool.
I've been trying to find a new family for years.
I'll be your family.
Oh, I love it.
The feel the love. So the unique species present in the cobra oral cavity was Bifidobacterium adolescentis. So the heat map indicates that most reads match the Cornybacterium being the most abundant at the genus level, followed by Bacteroides and Escherichia in the cobra, Physiococcus, Propionibacterium, Pseudomonas, and Mycobacterium in KC1. Mycobacterium, you know quite well. That is true.
Yes, I do.
Yes, that's
I'm wondering why that's there.
maybe it had tuberculosis. I don't know. In KC1, Fusobacterium providencia and Acinetobacter proteus and Bacteroides in KC2, and Escherichia coli and Physiococcus in the python. By comparing data from all snakes, it was found that E. coli, Propionibacterium acnes, Pseudomonas veroni, and Brevibacterium aureum, Serratium marcescens, and Morganella morgani were the most abundant bacteria at species level. Did you want to just say something? No. Okay. Wow. Yeah.
The fact that those are all like human pathogens, really. But the fact that the E. coli was in there, that tells you that it's coming from a prey item. And then for Serratia marcescens, that is a horrible human pathogen. So if they are surviving this just fine, I want to know what's in there. So really good idea for antibiotic research.
I thought Serratia marcescens was a fairly benign. Isn't that thing that causes the pink slime on your bathroom, or am I thinking of something else? Esma, I thought that wasn't a.
That could be Micrococcus luteus as well. Rosea.
Okay, because I thought Serratia marcescens was just like the.
Yes, it's definitely a pathogen. It's
it's an Enterobacter. Because it's bright. It's a bright red colony, isn't it?
Yep.
Ah. Ooh.
It is depending on the pH. Yes.
Ah. Okay. Right. Because I was reading an Answers in Genesis article about Serratia marcescens, how wonderful it was, and they used to eat wafers that was growing red because it was the blood of Jesus. And now you're telling me that Serratia marcescens is a a pathogen of the. Oh my god, that's hilarious. Oh wow!
I'm going to look this up. I have got to look this up.
Yeah, AIG, Serratia marcescens. Right. Okay. So you're saying that because they had Serratia marcescens, but they weren't sick, you're saying that it could have something to do with helping us with antimicrobial issues. Is that what you're saying?
Yeah.
Awesome.
They've got a natural antibiotic somehow.
Yeah.
Yeah. I mean, that's what I'm reading, and I'm thinking forward here.
Yeah.
I mean, it's slimy. So if it's in there, it's going to coat everything. There's going to be an enteric coating so the bacteria can get whatever nutrients are there. So how are they keeping it down?
I have no idea. See, this is why we do this sort of research, everyone. There is no such thing as bad research when it comes to science. Well, sometimes it is, but it depends on who's doing the research. Yeah.
Who's approving my grant is telling me that.
Because you mentioned something it's earlier, and I'll just go because it says here taxonomic analysis indicated that 50, 90, 93, 76, and 125 bacterial species in the oral cavities of cobra KC1, KC2, and python, respectively, and the majority of commonly abundant species were pathogen or opportunistic pathogens to humans. So yeah, you mentioned that because they were pathogens or opportunistic pathogens, so meaning that, like, again, we'll go back to thrush. Thrush is there, it just sits there, it's fine. But when you give it the opportunity to grow, for example, you suddenly have a high sugar diet or you're on antibiotics, which wipes out the lactobacillus, it now turns into a pathogen. So, yeah, the top 10 abundant bacterial species are listed in Table 2. Serratia marcescens, and I will go to the next one. Give me a moment. Most abundant, followed by Bacteroides fragilis, E. coli, and Propionibacterium acnes in the cobra snake. In KC1, the Pseudomonas veroni and Propionibacterium acnes were the most abundant, followed by Erwinia dispersa and Escherichia coli. Erwinia dispersa. That sounds like a Harry Potter spell. Erwinia dispersa. In descending order of their abundance...
I thought that was a really bad garage band.
That too. I can see that. Jassa G says, I've had Trent's mouth, which is very painful. Is that a thrush infection of the mouth? Because I've heard that's pretty nasty. In descending order of their abundance, the oral cavity of KC2 was occupied by Morganella morgani, Brevibacterium aureum, Bacteroides fragilis, and Propionibacterium acnes. Among the known species of the bacterial community, E. coli was the highly present, together with Propionibacterium acnes, Pseudomonas veroni, Serratia marcescens, and Brevibacterium aureum. It contributed the greatest share of the bacterial species in the python. The rarefaction plots elucidate that the python has more taxa, i.e. common 31 and unique 43, in the oral cavity as compared to the king cobra, KC1 and KC2 and cobra. And go to the next one. The alpha diversity result is shown in the additional file.
I want to add just one thing. These were individual snakes.
Pardon? Sorry? Yeah, these were the individual snakes.
These were individual snakes, so we don't know if that's for the entire species, if python does, or if it's really all of that species that have more than the venomous snakes.
Yeah, so you got to remember that this is an N value of 1. If you have an N value of 1, you know it's a pretty. It's not something that you should consider to be a Well, it's one of those ones that it's sort of like a proof of concept rather than this is exactly what's happened. But this is just interesting to say these are the results of our one Python. And it is kind of interesting, as you mentioned, that the Python had 1, 2, 3, 4, 5 different species. KC2 is 1, 2, 3. So maybe the venom within, like the venomous snakes, because you're saying that you pointed out just then that there's less variety of, or less commonality, I suppose you can say, of the cobras. I can't really. How does that look? Okay. If I show you that the Python. Okay, so this is your percentage of OTOs. Okay. All right. There's the infographic on one half of it. I'll see if I can. I have linked in the description below if you want to read along. I should have mentioned this earlier. The paper is in the description below. But I will go. I'll put it back onto. Okay, so you're talking about this, the specific species, because within the Python, you've got a lot of E. coli. And you've got 1, 2, 3, 4, 5, 6, 7, 8 different species in the Python, but the vast majority of it is E. coli. But if you look at the cobra, the K1, KC1, KC2, they're a lot more even out, especially with the cobra. If you're looking at the stream, that helps. Do you know where I'm talking about, or have I lost you?
It's cutting out really badly, so I can't keep up.
Oh, sorry. I've got a feeling my computer is... I don't think it's my internet. I actually think it might be my computer. It is very, very, very hot. So it's chugging away. So we'll try and wrap this up quickly. Okay. So the composition of taxa using stamps shows that Enterobacteriaceae, Corynebacterium, Enterococcus, Streptococcus, and Xanthomendaceae were significantly overrepresented, with positive difference in the 16S RNA gene amplicon surveys of the cobra, KC1, KC2, and Python. However, Moraxellaceae, Propionibacterium acnes, and Serratia marcescens were overrepresented with negative proportion differences. The p-values were estimated on Fisher's exact test. And I will... test method using Story's FDR approach. The correlations between cobra and king cobra shows that dominant microbes were positively correlated with P, less than 1e to the 15 significance. However, comparison to the cobra to the python revealed significant negative correlations in the abundance P1e to negative 6 of Enterobacteriaceae, Xanthomendaceae, and Streptophia. We'll go to this discussion. So very few studies limited to study of gut microbiota have attempted to characterize the bacterial organism colonizing the snake's gastrointestinal tract. Except for some culture-dependent studies, a complete profile of the oral bacterial community was absent in their species. Herein we investigated the oral bacterial community. composition of venomous and non-venomous snakes using the 16S RNA amplicon sequencing analysis. The present study demonstrated that within the phylum level, Proteobacteria and Actinobacteria were dominant in the oral bacterial community of the snake studies. And I think it starts talking about the sequence of captive Komodo dragons. And so it starts comparing it to other reptiles.
Right.
Yeah, so I might not read that because the stream's starting to die. But what's interesting, it does say a recent study demonstrated a dominant level of Actinobacteria and Proteobacteria in the oral cavity of a free-living passerine bird, the great tit. I just wanted to say that, really. The dominance of these two bacteriophyla in the oral cavities of snakes in our study suggests that snakes may show more similarities to birds as to their oral bacterial communities than to other vertebrate organisms. Like other creatures, snakes' oral cavity is a suitable place for bacterial growth, and some of them represent normal oral flora of animals in general. Studies on oral bacterial flora in snakes have been undertaken worldwide using culture-based methods, which is not very accurate because something like 99% of bacteria don't grow in culture. Different bacterial species have been recognized from the oral cavity of various varieties of snakes. The most significant ones are Pseudomonas and Eromonas, Morganella morganii, and Staphylococcus aureus, E. coli, Proteus colistridia, Enterococcus, Coagulase negative Staphylococcus, Dentotrophomonas maltophilia, Acinetobacter klebsiella, and Shigella. Shigella is really bad. That is not nice. It's a wonderful enterobacteria.
Klebsiella is a lung infection, and it can be fatal even with treatment.
Oh, wow.
Wow. Sorry, didn't mean to interrupt.
Yep. No, go ahead. Interrupt away. Staphylococcus, Salmonella, Escherichia, and Providencia. So they're just finding those particular species of microbes, and they're also finding that that this could mean it could lead to antibiotic development. Yeah. It says here the bacterial community in all the snakes were observed to be mixed population of the gram-positive and gram-negative organisms, and the commonly abundant bacteria were pathogens or opportunistic pathogens to humans. The wide spectrum of pathogens found the oral cavity of venomous snake studies whose bites may not cause not only give me a minute. Poisoning or envenomation, but also infection aggravates the condition from the victim. The literature indicates that bites from non-venomous snakes may also result in secondary bacterial infections. In this study, Propionibacterium acne, Serratia marsensins, and Ewina dispersa were the commonly found pathogenic species in the non-venomous python. Another thing I've noticed after reading lots and lots of scientific papers is that they tend to repeat themselves several hundred times.
They've got to make their point.
Yeah. Somehow. So subsequently, people associated with snake transportations or studies using these animals run the risk of being infected by opportunistic pathogens. The likelihood of infection is particularly high for persons who are sick or immunocompromised. All right, so I might actually, because what they're doing is just repeating themselves 5,000 times, I might just pop down to the conclusion. Okay.
While you're doing that, one thing that I would do that's slightly different in this is I would find out what's in the oral cavity by doing the swab and find out what's in the fecal material and see if something is missing.
Yeah. Yeah, I might actually end up doing that too, because that's something I might end up doing for my project. Okay, so the conclusion is, in this study, for the first time, we have shown oral bacterial flora in the venomous and non-venomous snake species from India using the next generation sequencing of hypervariable regions from the 16S RNA gene. Our study demonstrated a wide variation of bacterial species among these snakes whose oral cavities were predominantly occupied by both gram-negative and gram-positive pathogenic or opportunistic pathogenic bacteria. Our finding of a wide difference in Ophidian bacterial flora suggests a variation by individual species and geographical region. The results generated from this studies are of concern, as a bite inflicted by these snakes can result in wound infections and tissue necrosis, leading to sepsis, necrovising fasciitis, and or expose snake handlers, veterinarians, and researchers to infections. Furthermore, this work provides a foundation to carry out further research on snakes to recognize the potential drugs or antibiotics for treating different infectious diseases. That's basically it. After watching me get tortured by trying to say stuff. Yeah, so anything that you wanted to add to that? Anything you found interesting in that?
The fact that there are so many mammalian pathogens going through the GI tract of a reptile is mind-blowing to me. Those pathogens need to grow at 37 degrees. Like, those are the ones that, the ones that we commented on. Those are the ones that I typically grow in my lab if we're doing an experiment on something having to do with a human pathogen. Those are, those animals don't maintain their body temperature. So what do they do during the winter? What's going to happen? So then, is there a seasonality to the infection, or the because I know there's not a whole lot of seasons in India. I mean,
no,
there's months. It's either dry or wet. It's still hot,
but hot.
It's either hot and dry or hot and wet. Either way, but I mean, there are other reptiles, of course, in other areas of the planet. So I'm wondering if there's a seasonality to the type of bacteria that are visible, or found in the oral cavity, as well as the rest of the GI tract, or if it's constant. If it's constant, what keeps those pathogens, like ours? We've got an immune system, of course. What prevents those pathogens from affecting them the same way that it affects us? So there are I have more questions now than answers.
This is a new study for you now. You're like, I wonder if I can try and convince someone, because it would be really, really good if we could be able to take some things from the saliva of a snake and try and culture it. Because Staph aureus is really nasty if it's antibiotic resistant and infects places where it shouldn't be infecting. It's really nasty. It kills people. So yeah.
One of the bacteria that was also mentioned was the Mycobacterium, and you mentioned that as well. I mean, that's what I researched. Yeah. So if it's got Mycobacterium, it didn't say which one. I'm assuming it's I can't assume because I don't know. They didn't answer that question. But if it's got Mycobacterium, is that coming from the prey item that they're consuming, or is it something that's just endemic? Like, is it something that is constantly growing in the snake? If it's constantly growing in the snake, why isn't it affecting the snake?
Yeah. You're slightly excited about this, aren't you?
Yes, I am. I may have to travel to India very quickly.
After you get your vaccine.
I've already got it. I'm done.
Oh, you're done. Nice.
Oh, yeah.
You both? Well done. Slightly jealous.
All right.
I haven't been able to get mine yet.
We're sending them out as fast as we can.
Yeah. All right. Well, I'm going to let you go because I don't want to have everyone going, this is all choppy. So I will let you go. Thank you very much for joining me. I really appreciate it. I always love having you on. So, and thank you for coming on at just very, very, very short notice.
I am happy to be here. I love it. Absolutely love it. So I appreciate the opportunity.
And geeking over microbes is the thing we do best.
Yes.
All absolutely. Right. So where can we find you, by the way?
You can find me on Twitter. I'm on Twitter. I am Dr. Stephen Hobbs. You can look up the doctor or Dr. Stephen Hobbs. Either way, I'll be there. I'll answer. I try to interact with everybody as often as I can. So if you're there, you can send a follow, send me a message. Just say hello. I normally follow you back if you'll interact with me.
I will. So you're Dr. Stephen Hobbs on Twitter. I've put it in the chat.
Yes, the only social media I've got.
Yeah, it's probably a good idea too.
No, it's
all right. All right, and if you don't know who I am, I'm Meredith, aka Navadia. You'll find me on Twitter and my slowly dying YouTube channel. And anyway, but I just want to say thank you very much for coming and watching. It is a very fascinating subject. I'll try to get away from mouth microbes for next week. As I said, the hardest thing about this is trying to find short enough things. It's really hard because I said a lot of the things that I find is just incredible and I'd love to tell you about them, but they're 30 pages long, 25, 30 pages long. So anything less than 15 pages is pretty hard to find. So
endocarditis.
Pardon?
Endocarditis.
What's that? Okay, I'll look up endocarditis.
The inside of the heart. You get an oral microbe that gets introduced into the bloodstream. It goes to the heart.
Yeah, which is why you need to make sure you have good oral hygiene. So floss every day, which I don't do, much to the chagrin of my dentist. She's like, you need to floss. And I'm like, I keep forgetting.
It's been another year.
You should just be lucky I come. All right, I'm going to let you go. Thank you, everyone, for coming. And don't forget, don't give yourself the raw prawn or scandium iodine nitrogen serum, because that's the one that I should do at the end of the science one. All right. Bye, everyone.